Hello, welcome to the York Festival of Ideas. My name is Jenny Hayes from the University of York and I will be presenting today's DNA talk. Today I will be talking about the wonderful substance that is DNA, how we first discovered DNA, what it controls in our bodies, and how our bodies look after our DNA. I'll discuss how understanding DNA has allowed us to diagnose diseases and make biological medicines. I'll also cover some recent advances in sequencing and the implications this has for society. So, if there are any fellow knitters here, I won't keep you waiting. I decided to knit a DNA scarf a few years ago, and this is the result. I made one strand, then the second strand, then stitched the two strands together, then knitted a long backbone for each strand and stitched those on too. I'll explain what these letters along here mean shortly. The important principle from this slide is that trying to do complicated stuff without instructions will probably make a mess. When I started knitting, I followed instructions from other people, and I wouldn't have been able to create...
Could you speak up a bit? It's quite hard.
Sure.
Yeah,
I wouldn't be able to create the pattern for this scarf without having learned to knit first. Similarly, our bodies are very complicated, far more complicated than knitting one scarf. The instructions we inherited from our parents tell our bodies how to make us. Instructions that they inherited from their own parents, and so on, right back through billions of years of inheritance, back to the very first extremely simple single-celled organism at the start of life. So the information inside our cells that tells our bodies how to make a body is in the form of a molecule called deoxyribonucleic acid, or DNA. The entire DNA book of instructions for making an organism is called the genome. Now, our DNA is arranged in chromosomes where it's packaged up very tightly. Each tiny cell holding one copy of our entire genome has about 2 meters of DNA packed into it. This is far more impressive than my packing many meters of yarn into a scarf. If you were to zoom in onto a stretch of DNA on one of your chromosomes and read it, you would see the instructions for making lots of proteins. A gene is just a stretch of DNA that instructs the cell how it's going to make one protein. When your body does anything, it needs to use proteins that are all encoded in the DNA instructions. Enzymes are proteins that speed up chemical reactions in your body. Your digestive enzymes in your gut are all proteins. Your lungs breathe in oxygen, and this is carried around the body by a protein in red blood cells called hemoglobin. When diseases attack, your body uses these tiny little antibodies, that's more proteins, to defend itself. So how was DNA first discovered? Back in 1871, a Swiss physician, Friedrich Miescher, was studying white blood cells, which he got from the pus on bandages at the local hospital. Very gross. He was able to use a salt solution to wash the cells and extract the nuclei, and the precipitated substance from inside the nuclei was something that wasn't a protein. It was an interesting new substance, resistant to protein digestion methods, and it also contained a lot of phosphorus. One of Miescher's teachers, Albrecht Kossel, later isolated this and named it nucleic acid. He worked out the 5 components of nucleic acids over here on the side, and got the Nobel Prize for Physiology or Medicine in 1910 for this. So these 5 chemicals shown here are the nuclear bases that make up a nucleotide structure, such as DNA. We've got 3 bases, adenine and guanine on the far side, and cytosine. They're all present in DNA and the related material RNA, which is ribonucleic acid. Thymine is only present in DNA, and uracil is only present in RNA. Now, it's possible to extract DNA at home if you want to try. Here are some cups of DNA extracted from fruit. We've got banana and strawberry and kiwi fruit. You mash the fruit up, add half a cup of water, 2 teaspoons of washing up liquid, 1 teaspoon of table salt. Give all this a good mix. What you're doing here is getting the DNA to dissolve in water, which it quite likes doing. We're mostly water after all. Then filter the mixture through some filter paper to get rid of the solids. Then you add an equal volume of ice-cold surgical spirit or rubbing alcohol. You can get this at a pharmacy. the DNA doesn't dissolve so well in the alcohol, so it comes out of solution, it precipitates. So here I've stirred it with a toothpick, and you can see we've got white clumps of goopy precipitate. That's the DNA clinging onto the sticks in the two left cups, and really quite a lot of white DNA floating on the top in the right glass. Really good yield there. This DNA isn't quite as clean as in the lab, where we can use harsher chemicals and fancier equipment to get it really pure, but the principles are the same. So, by the early 1950s, biologists have worked out that DNA carries the instructions for life, and now the race is on to work out what DNA's structure is and how exactly it encodes the instructions for life. It's clear that these letters in DNA, A, C, G, T, must form a sequence, and that DNA has a regular structure. So James Watson, a molecular biologist, and Francis Crick, a biophysicist, have teamed up at the University of Cambridge, and they are designing physical models of how DNA might be put together. Meanwhile, in King's College London, Maurice Wilkins is testing DNA with a technique called X-ray crystallography. And this is where tiny crystals of a substance are blasted with X-rays, and this gives a diffraction pattern. You can see an example over here. If you use some complicated maths, it's possible to work backwards from the pattern and come up with some ideas for the structure that would have made it. So working out DNA's structure like this is a difficult problem and requires the best equipment available. A new researcher arrives, Rosalind Franklin, who'd previously done crystallography work on the mineral structure of coal. Her graduate student, Raymond Gosling, took photo 51, the one here on the right, showing a really clear diffraction pattern from DNA. And Franklin worked out some of the properties of the helical structure, a spiral shape, that would fit this pattern. Later on, as she prepares to leave King's College for Birkbeck, Maurice Wilkins gets her data and photographs. He shows Crick and Watson photo 51 and some data, and this gives them the final bit of the puzzle to deduce DNA's structure. So DNA, we now know, is this beautiful double helix form where two strands spiral around each other running in opposite directions. The bases are on the inside of the double helix, and as you can see on my scarf, they're matching up so that adenine is always opposite thymine, cytosine is always opposite guanine. So here I've covered the bases in with the colours I chose for my scarf, though you could pick any colours. 3 papers are published by the various people in April 1953. They outline DNA's structure and each group's evidence for it. As Crick had put it, they found the secret of life. After the structure of DNA was published, Watson moved to Harvard and started working on the related material, RNA. Crick continued to work on and how the DNA sequence codes for proteins. Wilkins continued to work on DNA as well. Gosling, who took the photo, went on to do research in medical physics. And Franklin worked on the X-ray crystallography structures of several plant viruses affecting crops, as well as poliovirus, but sadly died of ovarian cancer in 1958. Later on, the Nobel Prize for Physiology was awarded to Crick, Watson and Wilkins in 1962. So, our cells need to grow and divide to make new cells and replace old ones. And each new cell gets its own copy of our genome. This means that our bodies are always making more DNA. So when our DNA wants to replicate, it unwinds and unzips into 2 separate strands. I can do that with my scarf as well. Here you can see the strands are being pulled apart, and then an enzyme called DNA polymerase comes in. This is the type of protein I work on. DNA polymerase uses the bases already there as a template and adds the correct new bases onto each strand. So just here on the end we have a template thymine in green, so DNA polymerase will just put an adenine opposite like this. Next is adenine, so I'll put a thymine opposite. Guanine next, so I'll add a cytosine, and so on. And the polymerase goes along and makes a new strand, restoring the structure of a double helix. The new strand I've coloured in purple, so you get the idea. Over on this strand, we've got another DNA polymerase. Comes in and it's hard at work making a new strand. So we end up with 2 double helices that should be identical. Then the other proteins in the cell that handle DNA packaging can wind it all back up and package it back into a chromosome, and our cells have to do this for every chromosome every time they divide. So I mentioned that DNA carries the instructions to make proteins, so our cells need to be able to read and use the instructions. And the first step of this process is called transcription. So first, we'll unwind the DNA as before, and then another enzyme comes in. This time it's going to be RNA polymerase. This is going to make a single strand of RNA. I've mentioned RNA before, a similar substance to DNA with a sequence of letters coding for the gene. But RNA is usually just one strand, and I mentioned it has uracil instead of thymine, so all the letter T's become letter U's. So here the polymerase will add adenine, uracil, cytosine, and so on, and makes the new RNA strand. And then the DNA can get wound back into the double helix, get packed away. The RNA is going to go out of the nucleus into the cell and be read as the template for actually making the proteins. So just here I have the start of my scarf sequence, but I've put it in RNA form. So then we're outside of the nucleus in the cytosol of the cell, and a big structure called the ribosome comes along. So proteins like DNA are made from an alphabet of smaller components joined together. DNA has 4 bases, but protein has 20 different types of amino acids we're going to use. So here they all are, abbreviated and colour-coded for your convenience. The ribosome reads the RNA instructions and going through the sequence of letters 3 at a time. Each little triplet of letters, called a codon, tells the ribosome to add a single amino acid. So if you imagine I'm a ribosome, and my scarf's an RNA sequence, and I'm going to start reading, I read that this codon here, which in DNA was TTT, in the uracil it'll be UUU, there's a universal genetic code. And in that code, UUU means add the amino acid phenylalanine. So let's take one. Then I'll read the next triplet, the next codon, GUG. That codes the amino acid valine. So let's take a valine, join it on. Now the ribosome's going to move along and just read a third codon, so we'll read AAC, and that means add the amino acid asparagine and join that on. So the ribosome keeps moving along the RNA and makes a lovely long chain of amino acids called a polypeptide. They're all joined together. So let's make the polypeptide encoded by the entire DNA scarf that I'm wearing. Here it goes. Wonderful. If only making my scarf had been that easy. Now insulin is a very short protein. When first made, it is in a form that is 110 residues long. But then it gets made even shorter. After protein processing, just 51 amino acids end up in the finished product. Now, I'll admit I cheated a bit when designing my insulin scarf. In the interest of not having my fingers fall off, I didn't actually knit the entire insulin gene. Instead, I edited the original DNA sequence, hundreds of base pairs long, down to the 156 base pairs that code for the 51 amino acids. And then I separated those into chain A and chain B, which is how it's arranged in the final insulin structure. So this scarf that's 90 base pairs long is only part of a gene, the part encoding chain B of insulin. If I'd knitted the whole DNA sequence, the scarf would have been 5 times longer than this entire scarf. So, many proteins are also much bigger than this. Here's the entire insulin structure, chain A in red and chain B in blue. We've zoomed out so we can see it, and it's actually a tiny protein. An average protein is about 500 amino acids long. This big fat one is the protein alpha-2 macroglobulin. That's about 1,500 amino acid residues long. Titin is the longest protein in the human genome. That's about 30,000 amino acids long, depending on the RNA splicing. And sadly, I don't have time today to talk about RNA splicing and other interesting gene processing, and I'll certainly never have the time to knit a titin gene scarf, because that would be at least 1,000 times longer than the scarf I'm wearing. And yet each of us is, again, at the microscopic level, constantly doing this many times over in every one of our cells, making the proteins we need. So the finished proteins do whatever job they have in the body. They digest food, carry oxygen, fighting intruders, all the stuff we need to do to survive. And we need the millions of ribosomes we have in each cell to work all the time, because if our body ever stops making new proteins to replace the worn-out ones, we face a rather grisly death. This is the danger of eating the death cap mushroom, which can kill you with just half a cap. This mushroom contains a deadly poison called alpha-amanitin that stops RNA polymerase from reading the DNA and making the RNA intermediate. So then, no new RNA is made, which means no new protein is made either. Oops. About 10 to 24 hours after eating the poisonous mushrooms, the unfortunate person is going to start feeling quite ill. This lag time is very bad because it's too late to pump their stomach at this point, and there's no known antidote to the alpha-amanitin poison. Modern medicine has improved survival rates, but as liver failure takes hold, the patient may urgently need a liver transplant to survive, and some patients don't make it. Now, this very deadly poison is being trialled as a possible chemotherapy treatment for cancer. I gave a talk at last year's festival on how do plants become medicines. One of the things I mentioned in that talk is that the dose makes the poison. To stop the poison from overdosing healthy cells and killing the patient, we're going to have to use it as an antibody-drug conjugate. This is a really neat type of chemotherapy where an antibody, a protein I mentioned earlier that fights disease, is selected that will recognize tumour cells. Linked onto the antibody is a drug, a powerful poison. In this case we're using alpha-amanitin. Essentially we're using the antibody as a delivery system that will target the amanitin to tumour cells. Here, take this package please, and hopefully mean that only those cells get the fatal dose. So there are 3 billion base pairs in the human genome, and that's a very long string of instructions. When I made my scarf, I edited the sequence. As I said, I wanted it edited down to the most interesting bits because that was all I needed. Similarly, although almost all our cells have a copy of our whole genome, there isn't a single cell that actually reads all the instructions in our DNA. They don't need to, they just need to read the bits that are important to them. So the cardiac muscle cells read the instructions for titin, the extremely long protein I mentioned earlier, plus a bunch of other muscle-related proteins. The brain cells reading a different set of instructions, making the proteins that turn it into a neuron. The bone cells reading another set of instructions, another set for the white blood cells in the immune system. Sometimes cells read the same instructions. All cells need food, for example, so they're all reading the chapter on how to get energy from breaking down glucose. Sometimes they're reading different chapters, and this is what makes a complicated organism like a human, where we have lots of different cell types. So our cells read the DNA instructions in order to make all our proteins. But this whole process relies on the instructions being correct. DNA is under attack all the time from oxidation, reactive chemicals. We've got ultraviolet light and ionizing radiation coming in. There's also replication errors. Even if DNA was shielded from every source of damage and wasn't doing anything, the components will break down over time. So this means that our DNA is constantly undergoing mutations. Our cells have to be able to reverse this damage, otherwise the instructions for making proteins will be all wrong. So imagine I'm reading a book on how I'm going to make a cell and put some spikes on it. Except I find a typo in the book. It says to put spokes on the cell. Well, if I know that's not right, I can Tipp-Ex over the word and then write it correctly with a pen. So it's fixed, and the instructions are now for adding spikes. I could even do this to correct a whole sentence if I knew what the sentence was supposed to say. Or maybe there's a page torn out of my instructions and I really wanted to know how the book ends, so I seek out another copy of the book to go and read the missing page. Wow, what a twist. I can even photocopy the missing page and glue the new copy back into the first book to fix it. And similarly, all living things have DNA repair proteins that can detect and fix some forms of DNA damage. During DNA replication, a mismatch might occur when DNA polymerase adds new bases. So here, an adenine wrongly sits opposite a cytosine. There's a proofreading section of the polymerase that goes, "Hold it, no, that's wrong. Remove it and try again." And then it can add the correct base. Large chunks of damaged DNA over on the strand, like the mess over here, this can be cut out by repair enzymes. So we remove the damage, and then polymerase comes in and makes a new strand to replace it. Even serious damage to both DNA strands can be fixed by a process that takes advantage of the fact that our chromosomes come in pairs. The intact chromosome is used as a template to accurately fix the broken chromosome, in the same way that I can photocopy a second copy of a book to repair the damaged copy, we can repair the broken chromosome. So our cells have multiple ways of dealing with DNA damage. They're generally pretty good at fixing mutations, but they're not perfect. As living beings, we slowly accumulate DNA damage over time, which contributes to problems such as aging and cancer. It's also possible for DNA mutations to occur in egg and sperm or at the embryo stage, and this can mean an entire organism has a mutation in every cell in their body. So this is a genetic disorder. There are thousands of genetic disorders documented in humans, some of them fairly common, so they affect 1 in a few thousand people. Other diseases are so rare, they've only been documented a few times. The first genetic disease that biochemists figured out is sickle cell anemia, a condition most common in tropical regions. Under the microscope, you can see the red blood cells, which are normally this nice round shape, become sickle-shaped. The main symptom is the sickle cell crisis, a painful and dangerous condition where the abnormal blood cells are clogging up the capillaries and preventing blood flow to the organs beyond. And this organ damage can be fatal. Untreated patients aren't likely to make it to adulthood. Even with modern treatment, life expectancy is only into the 60s. Now, Linus Pauling in 1949 noted that hemoglobin in the sickle-shaped cells aggregates together. It forms these clumps when oxygen levels are low. And he speculated something had happened to the hemoglobin at the molecular level. Frederick Sanger develops a method of sequencing protein in the 1950s and gets a Nobel Prize for this. It's now possible to go through the amino acids making up the chain and identify each one. And in 1957, Vernon Ingram locates the mutation causing sickle cell. In the beta chain of hemoglobin, a single glutamic acid substituted for a valine is the one change that causes the hemoglobin chains to stick together and aggregate into these long strands that distort the cell into the sickle shape. So all genetic diseases come down to this principle. We have a specific mutation in a protein that changes its structure, which then changes how it functions and causes the disease symptoms. But it's taken a lot of work to get to this point, and scientists want to pin down exactly what happened in the DNA and figure out a good way for them to diagnose patients quickly. So an important technique in the lab, which I use regularly, is the ability to make lots of copies of a single bit of DNA. We've seen cells make new copies of their genome by using DNA polymerase. In the 1980s, Carrie Mullis developed the polymerase chain reaction, known as PCR. So I'll have the gene we're studying, some carefully chosen primers. These are very short bits of DNA that correctly match the start and end of the gene. We've got plenty of fresh bases for the polymerase to use, and of course we'll have a polymerase to make the new DNA. So the first step is to heat the DNA to 95 degrees, and this melts the DNA. Essentially the two strands just fall apart. Now as we carefully cool the DNA down, the primers will go and match up at the correct points, and then if we add some polymerase in, it can bind on at the primers and make some more DNA. So that's wonderful, we've doubled the DNA to 2 strands. We could even do this again. If we heat up the DNA and melt it again, oh no, what's happened to the polymerase? We've ruined it! Polymerase is a protein. It doesn't like heat. If you imagine what happens to an egg when you fry it, that's protein being denatured. by heat. The early versions of PCR required people to add fresh polymerase after every single step. This was very time-consuming, very labour-intensive. It meant that it was very easy to contaminate it on just one of the steps, and if that happens just once, by the end we're going to get a mess. So, to avoid the mess, it's time to go somewhere nice and warm. So, this place should probably be hot enough. If you go to volcanic areas, you can find a lot of awesome stuff, and I love the colours you can get around volcanoes. A vivid reddish-orange lava eruption, the yellow sulphur mines, even the so-called blue lava of Kawa-Egen. This is actually a burning sulphur field. A few places have all the colours, and one of these, a wonderful place, is Yellowstone. This is the Grand Prismatic Spring, and it was discovered that the colours round the edges are due to the bacteria that live in the hot water. The most heat-resistant ones live closer to the centre, and the slightly less heat-resistant ones live further out. So in the 1960s, Thomas Brock was one of the first microbiologists to investigate hot springs in Yellowstone, and isolated the bacterium Thermus aquaticus from a place called Mushroom Spring. Now, Thermus aquaticus grows handily at 70 degrees Celsius, and at the time it was the hottest temperature known that life could survive at. So the discovery of a new thermostable polymerase, one that isn't degraded by heat, meant that PCR became much more useful, because now we can do the chain reaction all at once without having to stop and add new polymerase each cycle, without risking contaminating it, messing everything up. So we take our starting ingredients, including the new thermostable TAC DNA polymerase. The enzymes are named based on the bacterium, so TAC denotes Thermus aquaticus. We'll heat the DNA to 95 degrees and melt it. We'll cool it down to around 55 degrees, and the primers will come over and attach. We'll heat it up to 72 degrees, because that's the temperature TAC polymerase likes to make some more DNA. So the polymerase comes in, and there we go. That's one cycle that has doubled the DNA. And now we just repeat the whole process. We'll heat again to 95 degrees, which doesn't damage the TAC. It can tolerate the temperatures that high. We'll cool it down and attach some more primers. Heat up the polymerase so it gets to work at its optimum temperature. Boom, cycle 2, and we've doubled to 4 copies of the DNA. So let's keep going. If I run 3 cycles, I'll be up to 8 copies of DNA. If I do 4 cycles, oh boy, the DNA is about to spill off the end of the slide here. I'm greedy, and that's not enough DNA for me. In the lab, when I run a PCR, I like to do 30 cycles. So in theory, I'll get over 1 billion copies of the DNA. That should be enough. Although in practice, the process is not 100% efficient, so we'll probably end up with several million copies of DNA. But with PCR, scientists can essentially make as much DNA as we want. Now, I'm working on looking for new polymerases. Since the discovery of TAC, many other bacteria have been found that thrive in hot springs and around deep water volcanic vents. If we're going even hotter, it means we get a polymerase that can survive the high temperature step of PCR for longer, so then we can run it for more cycles. And also some bacteria have polymerases with other interesting properties, such as proofreading and repairing tricky DNA damage. So these are some of my favourite bacteria. We've got Thermus aquaticus, of course, a classic. Cod is a polymerase I use quite a lot. Thermococcus codicariensis comes from a steam vent off the island of Kodakarajima, Japan. This tolerates 85 degrees, no problem. Pyrococcus furiosus, what an awesome name that is, means rushing fireberry. It refers to its furiously fast growth rate. This one can easily survive temperatures up to 100 degrees Celsius, and it was discovered in Volcano, Italy. And finally, a really weird one down in the corner, Dinococcus radiodurans, discovered in 1956 by scientists trying to work out if gamma radiation could be used to sterilize food. Now, this beastie can survive radiation 1,000 times stronger than the dose that kills a human. The radiation shatters its genome into dozens of pieces, and yet it has some wonderful DNA repair proteins that can put all the pieces back together without a single mistake. It's also resistant to acid and dehydration, and survived a 3-year experiment sitting on the outside of the International Space Station. So, polymerases are great. Another type of tool for manipulating DNA was discovered in bacteria in the 1950s. Bacteriophage here are a type of virus that infect bacteria by inserting their own DNA, but they don't always do this successfully. Organisms can mark their DNA in certain ways to identify it as DNA that should be there, and that way if some unmarked DNA suddenly appears, it doesn't belong and it can get sliced up and removed by proteins called restriction enzymes that act like a pair of molecular-sized scissors. So the cell is restricting the access of foreign DNA, going, hold it, you shouldn't be here, and attacking it like a very small immune system. The handy thing is there are lots and lots of bacteria infected by a specific type of phage that have their own specific restriction enzyme that they defend themselves with. When scientists realised that these different enzymes only slice at certain sequences, called restriction sites, we suddenly had a new toolbox to play with. So perhaps I will slice this sequence. I'll use some SMAR1 from Serratia maricens. There we go. Or for this sequence over here, perhaps some HindD3 from Haemophilus influenzae will do the job. There are thousands of these enzymes now discovered from screening many bacteria. Using these precise tools, we can basically slice up DNA at will in the laboratory. And we can even stick pieces back together using another enzyme called DNA ligase, which seals the overhanging ends back together. So with all these new tools, it became possible to diagnose sickle cell disease much faster. First, we'll use PCR to make 200,000 copies of the DNA. Then we'll carefully select a couple of restriction enzymes that recognize the sites around the mutation. And these will cut the normal beta hemoglobin sequence one way and cut the sickle version a different way. So then if we run the DNA on a gel to separate the pieces according to length, we can see a difference. The DNA sequence from a healthy person looks like this. Over on the other side, we've got the DNA from someone who has the disease, looks different. And now any future patients being screened, even as newborns, can have their DNA tested by PCR and just compared to this reference. There's also sickle cell trait, which is the term for when one person has inherited one faulty copy of beta hemoglobin and one healthy copy, which means that both sequences appear. Now these people usually don't have the disease symptoms, and an interesting effect is that they're more resistant to malaria, and this is why the sickle cell gene hasn't been selected out in tropical regions. So PCR has been applied to many diseases since. You remember COVID testing? People took a lateral flow test, and if it was positive, you had to take up a follow-up test to confirm if it was definitely COVID. That was a PCR test. And this allowed us to keep track of which strains were emerging, like the Omicron strain, how many people were being infected by the new strains. There are lots of other diseases. We've got viral ones, influenza and HIV, bacterial ones such as strep throat and tuberculosis. Any infectious disease can be detected by PCR. This allows us to diagnose patients and select the ideal treatment based on the pathogen in its strain. And then there's even more uses of PCR, genetic fingerprinting for paternity testing or crime scene investigation, testing prospective parents to see if they're carriers for a genetic disease, testing tissue matches for organ transplantation. We can test cancer biopsies to see what mutations are present in the cancer and therefore what drugs might work. Herceptin, for instance, a drug used for breast cancer where it's mutated and overexpressed the HER2 receptor. And similarly, if gastric cancer has a VEGF mutation, we'll use a drug to target that. And then another important breakthrough was DNA sequencing. I mentioned Fred Sanger once already. His invention of the Sanger sequencing method for DNA in 1977 got him his second Nobel Prize. Busy man. In the Sanger method, we have a strand of DNA, and we're making a new strand opposite using polymerase. Much like a PCR reaction, but here, if we add a particular type of base called a dideoxynucleotide, it matches on correctly, but then stops the synthesis going any further. So we end up with all these strands of different lengths, depending on when they were halted, and if you use fluorescent dideoxynucleotides, these strands will glow different colours when exposed to laser light. So if we separate these out through a capillary and read it with a laser, it gives a beautiful readout. where we can directly see which letter is at each point on the DNA. I think there's a stall out in the atrium where you can see more about this. So Sanger sequencing was used to sequence the original Human Genome Project, which took 13 years and cost around 2.7 billion. Now, sequencing technology has advanced so much, we can do a whole human genome in a few days and cost less than $1,000. Sequencing DNA allows us to know exactly what the DNA says. Now, this doesn't mean that we know everything that's important. The process to RNA, and then to protein, and then the billions and billions of interactions between proteins in our cells, all adds a lot more complexity on top of that. So there are limits on what knowing a DNA sequence can tell us. But sequencing DNA has got many medical applications, testing for many human genetic diseases, of course, sequencing infectious diseases like COVID to track different strains, sequencing cancers to figure out what's going on with the cells, what drug treatments might work. There can be good medical reasons to sequence people preemptively. If people have a family history of breast cancer, they might want to check their DNA, and if they find out they've got the BRCA1 mutation, they can take preventative treatment before the disease develops. But cheap sequencing also throws up some questions about what else you can do with your DNA. People with sickle cell disease are also more easily damaged by exposure to nitro and amino compounds, and for this reason chemical manufacturing companies began screening for sickle cell as far back as the 1970s. Should an employer know about an employee's genetic makeup? What does that mean for the employee if their health insurance wants to know this as well? There's plenty of people who sent their DNA off to 23andMe, Ancestry and other similar companies out of curiosity. I've toyed with the idea myself. But 23andMe filed for bankruptcy in March of this year, and there are questions about what's going to happen to the DNA data. Entrusting capitalist companies with your DNA runs the risk that at some point they will find a way to profit from it, and will do so even if it hurts you. And even if one person is happy for their own genetic information to be shared, if they do so, it's tantamount to sharing the DNA of their relatives as well. So we'll go back to medicine. If we use all our tricks that we've now got in the DNA toolbox, we can make DNA for our own purposes, such as manufacturing biological drugs in industrial quantities. We'll start with this plasmid. This is just a circular loop of DNA separate from the main chromosome found in bacteria. We'll take the plasmid, cut it open with those restriction enzymes, take a gene of something interesting that we've cut with the same restriction enzymes. We'll mix them together and use ligase to stitch the DNA together, and now the plasmid has the gene coding for our wanted protein. And if we get a suitable strain of E. coli to accept the plasmid, E. coli will start making us lots of the protein. We're essentially giving E. coli new instructions. So, we can start with some insulin, make that for us E. coli. That's a medicine all type 1 diabetics need, and before being able to make genetically engineered bacteria, insulin had to be extracted from pigs and cows. Aside from medical applications, there's also food. Cheese making on an industrial scale requires enzymes that we can make in genetically modified fungi. We also aren't restricted to modifying microorganisms. We've advanced far enough to develop some genetically modified plants. So golden rice, for instance, a genetically engineered version that's high levels of beta-carotene, which we can convert to vitamin A. All crops can be made resistant to herbicides or pests. In the lab, one of my favourite research applications of this is to modify cells with fluorescent proteins, which allows us to see what's going on at the microscopic level, and also looks really cool. This is a painting from the Roger Chain lab, made by streaking 8 strains of bacteria, all expressing differently coloured fluorescent proteins. There are heaps of biological drugs. If it's a large biological structure and difficult to make it chemically, we just tell E. coli to make it instead. I had a check of the top 10 drugs of 2024 by sales. Antibodies are a bestseller. 5 of the drugs are antibodies, including the number one, pembrolizumab. All antibody drugs, by the way, end in mAb, which stands for monoclonal antibody. The technology has really advanced over the years. It started with making mouse antibodies against whatever protein you wanted to research in the lab. But with successive genetic slicing and stitching, we've made chimeric antibodies that combine the red mouse regions here with a human base in blue. We've got humanized antibodies that are 5% mouse and 95% human. And finally, we've developed fully human antibodies, but they're made in genetically modified mice carrying human antibody genes. So the original mouse ones are only now used in diagnosis in the lab, such as detecting HIV, because each successive generation is less likely to provoke a dangerous immune response. Two more of the top 10 drugs of 2024. We've got weight loss polypeptides like semaglutide, which has increased in popularity in the last couple of years. So just in the top 10, we have 7 biological drugs being used for chemotherapy, metabolic disorders, and immunology treatment. There are many other medical treatments. Insulin I've mentioned. Factor VIII for haemophiliacs, the previous method for this, pulling plasma from thousands of donors to extract the factor VIII, led to the contaminated blood scandal. If you make a virus protein, such as hepatitis B, you can stick that in a vaccine, or you can just make any protein at all to study its structure in the lab. So the last technique I want to talk about is CRISPR. The CRISPR-Cas9 system was discovered in the flesh-eating bacterium Streptococcus pyogenes. It's another way for bacteria to defend themselves against phage. The bacterium carries some RNA that's the genetic sequence from a phage from a previous infection. And when a phage of the same variety injects its DNA, the matching RNA here in green locks on to the DNA and allows the purple Cas9 protein to target the invading DNA, slicing it up and stopping the infection. And this system has been modified to target and slice genetic sequences in cells. It can even, after slicing the DNA open, add a new sequence in and stitch it back together. And this powerful technique got Jennifer Doudna and Emmanuelle Charpentier the Nobel Prize for Chemistry in 2020. So with CRISPR-Cas9, we're on the verge of being able to cure genetic diseases. Sickle cell anemia, for example. Our bone marrow contains stem cells that make all the blood cells in our bodies. It's possible to undergo a bone marrow transplant, a grueling treatment where chemotherapy is used to obliterate the stem cells in a patient's bone marrow. They then receive new stem cells from a donor matched as closely as possible, and then they have to take immunosuppressant drugs to make sure their immune system doesn't attack the foreign cells. Usually a family member offers the best chance of a good match, but a match can come from anywhere, although many people with leukemia and other life-threatening diseases die before finding a match. I would encourage anyone to consider registering with one of the bone marrow charities. I myself signed up a few years ago. The odds of you being called up for a donation are very small, but you just might be the perfect match to save someone's life. And if you are called up, the process is usually not much more difficult than donating blood. Now, for a leukemia patient, their options are likely to be get a successful transplant or die. For sickle cell anemia, a bone marrow transplant is usually a last resort. The effects of sickle cell disease might kill the patient, the side effects of the transplant might kill the patient. It's a little hard to know what's best. And CRISPR means there's a new treatment available. So this new method is to take stem cells from the patient's own bone marrow, use CRISPR to switch off the faulty beta hemoglobin gene, and this forces the stem cells to switch to making gamma hemoglobin instead, which your body makes when it's a fetus. And gamma hemoglobin isn't affected by the mutation. So when the modified cells are put back in the patient, they start making the fetal hemoglobin, and the patient's symptoms are cured. And it also means you don't risk graft versus host disease because the cells originally belonged to the patient, so they're already a perfect match. So this is a seriously life-changing cure, and I'm looking forward to this being rolled out to other genetic diseases. Beta thalassemia, that's another blood disorder that's an obvious candidate. Fatal genetic diseases, Huntington's, muscular dystrophy, cystic fibrosis. There are lots of diseases that in theory can be cured by modifying the damaged gene. And this includes lots of individually rare genetic diseases that have no hope of a drug being developed for them due to the tiny market for such a drug. And these might be cured with a standard CRISPR-Cas9 system where the RNA sequence joining on is the only component that needs tailored to each patient. So, now we're coming to the end of my talk. There are 3 billion base pairs in the human genome, of which this is a tiny, tiny subset. We've read them all many times now. But we understand only a small part of what the DNA is coding for and what the resulting proteins are doing. Some of the mutations that can occur in DNA result in some very nasty diseases. And over the years, we've developed a lot of advanced techniques for looking at and modifying DNA. So DNA research is a very exciting place to be right now as we begin to roll out the first gene therapies. And meanwhile, if anyone does want to try making their own DNA scarf, the scarf for insulin chain B is the one I'm wearing. And you can also make a slightly shorter scarf for insulin chain A. So there's the instructions up here, how you put the scarf together. I basically knitted 4 separate scarves, all quite thin, and then stitched everything together. And I have a few printouts of the instructions down here as well, should you want them. So that's it. I have drawn many diagrams for this talk, but any diagrams or pictures not made by me have been taken from the good people at Wikimedia Commons. These pictures are all copyright free, either public domain or used under a Creative Commons license, requiring only that I credit the author. So my thanks to all these people for their time and resources. So that's the end of my talk. Thank you for attending, and be sure to check out what the next week has in store in the festival programme. And we have some time for questions now, if anyone wants to ask me about DNA, biochemistry, working in research, or even knitting. Thank you.
Thank you. Yeah. Well, I know all about polypeptides because my mum used to do it for tape. She used to tape for the blind for Open University in the 80s. So she knew all about polypeptides and all this sort of thing. She worked at Shell. So, yeah, very, very interesting. And how big are these? What's the size of DNA? What?
Oh, the size of DNA. The size of DNA is very, very tiny.
In terms of millimetres?
In terms of millimetres, probably a few millionths of a millimetre. They're that small. There's a unit called an angstrom, which is you can fit 10 billion angstroms in a metre or something. So yes, when we're dealing with things down at the molecular size, this really is tiny.
What millionth of a millimetre? Yeah.
Yeah, a millionth of a millimetre is probably about the right ballpark.
Yeah, cheers. Thank you. Very, very interesting. Thank you very much.
I remember when reading about DNA several years ago, it was observed that there were large dark regions in the genome, and I wondered if that was still the case, or if now all the parts of the genome are known, their role in genetics is now understood.
There's a yes and no to that. We have read just about all the genome now. So I mentioned the Human Genome Project. When they published the sequence, it was only actually about 92% complete. I brought you up a picture of some chromosomes. You have this sort of X shape of a chromosome, and the bit in the middle in particular is this repetitive packed up sequence that's really hard to read, and there's a couple of other regions that are quite difficult. So when they published the Human Genome Project, the first version, it was only actually about 92% done. Now, I think we're about 2011 or 2012, so a decade or so afterwards, they finally filled in the last bit, so we can now honestly hand on heart say we have read every letter of the genome. So yes, we've done that.
But that wasn't what
in terms of the next bit, asking do we know what it's doing? No, we don't know what it's doing. There's a lot of bits of the genome that are still, I'll look through genomes of things, bacteria at work, for example, and a number of things which will just be listed as hypothetical protein. We know that there's something there. We've got a start codon, some sequence, an end codon, so that's probably coding for something. But so much of this stuff still hasn't been made, still hasn't been investigated, still hasn't been characterised. So there's a lot of work still to do, which at least keeps me in a job.
Thank you. Your depiction of the DNA polymerase acting has focused on the complementarity of the bases, but the sequence is actually determined only because the deoxyribose is linked through phosphates all the way. That's the hem of your scarf. And at what stage does that happen? Does the DNA polymerase actually present mononucleotides? That's to say, the base already attached to the sugar. Yeah. It does, but then the phosphate is crucial to the sequence, but that's the last step, is it?
Well, you've got they usually ATP, CTP, GTP, and TTP. So those are the thymine trinucleotide phosphate, for example. So we've got thymine triphosphate. So basically, when the DNA polymerase takes the component it needs, I depict it as just the sort of bit here, the colourful bit, but actually it's sort of got the backbone already attached. We've got the base, the letter that we need. We've got the sugar, either ribose for RNA or deoxyribose for DNA. And we've got some phosphate groups, 3 of them lined up, already attached. And these building blocks are put together by other proteins in the immune system. So then when we've got the building blocks together, essentially the building block will come up to the polymerase. The polymerase breaks off 2 of the phosphates. It'll use the energy in breaking those off for the next step, because it needs the energy to power the step. And then we've got the letter, the sugar and the phosphate all still joined together, which is jammed in and attached. So that's the bit at which it actually adds the letter and continues to make the stem.
Thank you.
You mentioned at the beginning that the genome forms a helix.
Yes.
Does this have any significance? All the diagrams you've shown as sort of zip diagrams,
yes,
rather than forming a helix.
Yes, I show it as a zip diagram because it's easier to see what the bases are doing.
Yeah.
In the as we see from the X-ray diffraction photo, in practice it's usually wound around into a helix.
Yeah, but does it have any significance?
I think the main significance. Yeah, I think the main significance of it is just the packaging. If you twist it around, then the way the molecules fit together sort of neatly packs together more easily if it spirals around.
Yeah. Just what chromosomes and genes, given the genome, a sequence of, is that 4 letters?
Yeah, there's 4 letters.
Yeah, if you're given a whole sequence, how would you split that into genes, and how would chromosomes fit into that pattern?
Right, so the chromosomes are units that we can physically see down a microscope.
Yeah, so
genes usually aren't spread across chromosomes, they'll be on one chromosome.
Yeah.
So you'll go down to chromosome level, within the chromosome you'll have thousands of genes on it. The human genome has about 23,000 genes. So we've got 23 pairs of chromosomes, bigger and smaller ones.
Yeah, so this split in a different chapter, so to speak.
Yeah. And then in terms of getting a gene, I mentioned the 20 amino acids.
Yeah.
Proteins always start with a methionine, handily enough, and then there's a few codons in the universal genetic code that mean stop.
Yeah.
So I mentioned where I can read through the bacterial genome, and the genome can be marked. Previously it was done by a human having to read through the letters, poor sod. Now we can have the computer read through it and go ATG, that's a methionine and therefore a potential start point. UAA, that's a stop codon, so there might be a stop there. And then you can label, here's a gene.
So the gene structure has punctuation marks?
It does have punctuation marks, it has capital letters, it has full stops.
Yeah, okay, thank you very much.
My question is more practical.
Okay.
For the treatment of leukemia, you talk about the CRISPR technique.
Yes.
Is it still at the research stage, or is it used at the moment?
It's starting to be rolled out for sickle cell. I'm not 100% certain if it's been rolled out to leukemia yet. You could have the same thing in theory. It might be more difficult. The deal with sickle cell and why it's started to be rolled onto the NHS as of, I think it was November last year was that headline, is that sickle cell is traced to just one mutation. One single letter has changed, one single amino acid has changed. So we need CRISPR to perform one error correction, and then we can roll it out into the body. Now with leukemia, the trouble with cancers is they usually start making quite a lot of mistakes in DNA replication. There's certain genes that sort of help proofread the genome, the DNA repair proteins that help keep it good, and these often become mutated in cancers, including leukemia. So in terms of leukemia, the current treatment is still to find someone else who's a good match for them. Now in theory, I think you could sequence a person's normal cells, sequence a leukemia patient's bone marrow that's got the cancer and has gone wrong, go through it and collect every single mistake that's been made, and then figure out a way to CRISPR some stem cells, correct each step very carefully, and then put fresh stem cells, perfectly good stem cells, back into the patient. So I think in theory it could probably be done. I don't know if we're trying it in leukemia patients yet. It'll probably be a more difficult thing, but the thing about genetic modification is there are upsides and there are downsides. If you're facing death, you're willing to take more risks, whereas if you're not facing death, then you want to be more cautious about editing the genome because you might make something go wrong.
Okay. Thank you.
Okay.
Thank you very much for your description. Obviously a very complicated world. Can you give us any guidance about how on earth this world evolved? Because clearly it didn't start like this.
I can have some ideas. So there's a particular hypothesis called the RNA world hypothesis. The idea is that we have, I showed you the steps of what's called the central dogma, where DNA goes to RNA goes to protein. But that's a simplified version of what most commonly happens in our bodies now. There are other processes I didn't talk about much, and maybe they were more important in early life. So basically, we think RNA, the middle step, evolved first. Before the first step of DNA, before the last step of proteins, we think that actually there was a time, the RNA world hypothesis, when life was just RNA molecules. So RNA has the ability to turn around and replicate itself, and can also sometimes fold itself up into structures and then sort of behave a bit like a protein, a bit like an enzyme, and cut up other things. So in theory, you could have an RNA that does two tasks, that it does something to digest a sugar or something to make some food, and can also turn around and replicate itself. Such a simple system would, in theory, be, I don't know, would you call it alive? There's a really neat experiment where they took a ribosomal genome, I can't remember how long, it was something like 5,000 letters to begin with, put it in a lab and basically put it under extreme pressure to mutate and to reproduce as fast as possible. Now if you're small, you reproduce faster than something that's big, so they were trying to force this genome down to be as small as possible, and they ended up with this sequence of RNA, just 200 letters long, that could replicate itself. They called it the Spiegelman's monster. So we think that possibly, it's difficult to go back and check 4 million years ago, but 4 billion years ago, but we think that that life started with RNA sequences arranging themselves into something which isn't particularly good at storing information, that's why DNA later evolved, isn't particularly good at doing the range of reactions that proteins do, that's why proteins later evolved, but that was the first step. Then later on, organisms can start to use DNA and proteins. So that's an interesting subject. We could have a whole talk on ancient bacteria.
What was it called?
Spiegelman's monster. Yes.
Monster.
So yes, I don't know, is Spiegelman's monster alive? It feels like it's really stretching the limits of life, and yet it replicates itself, and it is biological.
How often does this DNA get replaced in your body? How often? When it splits to a new cell?
Oh, replicating. Well, yeah, when your cells split, obviously your certain bits of your cells, like your lining of your nose and your lining of your gut and stuff, are replaced every few days. Yeah. Well, yeah, your gut lining only lasts about 72 hours. And various skin cells obviously get replaced quite regularly. Some cells are a lot more longer lived. I think your bone cells get turned over once a decade or so, and some brain cells can obviously last a lifetime, so they're not dividing very much. But yeah, you need to make our bodies always need to make a lot of DNA, and also repair DNA. I mentioned how even by itself DNA will break down without regular housekeeping by the repair proteins. Right, I've got some more questions over there.
You used the expression at one point, the human genome.
Yes.
But my DNA differs from yours, and DNA nowadays is used for identification of individuals in populations of millions. Can you briefly clarify what appears to be a contradiction?
Yeah, so if I say the Human Genome Project, for example, I'm referring to an amalgamation of, I think it was 8 individuals who they sequenced. Some things are the same across people, like we've all got the same gene for hemoglobin, hopefully, unless we're sick with sickle cell. And there's lots of other proteins in the body that are supposed to have that one function, and either they have this one sequence and they function correctly, or the sequences are on, in which case it's a mutation and it's not working, in which case we know that's wrong. So there's a lot of proteins in the body there's a lot of DNA sequences in the body where we know what it's supposed to be. There's equally many sequences. You mentioned, I think, a single nucleotide polymorphisms, for instance, are a good example of this. They're almost like ID markers. They didn't really evolve as ID markers. It's speculated that they're junk DNA that may derive from ancient viruses that integrated into the genome. But they're very useful because they often, as you say, differ between individuals. And so we can use them to identify. So as you say, each person's genome is actually unique. if I refer to the human genome, I'm referring to the consensus genome rather than a single sequence which is correct, as it were.
Are you really saying, as part of the answer to that, that most of our genome is identical and it's little bits that are different?
Yes.
Yeah.
I would say, people say we're 98% chimp or whatever it is, we're 99.9% human, all of us. We only have a very small amount that differs. But that amount that differs is still important because it stops us all being identical twins.
And helps to fight criminals as well, doesn't it? DNA.
Yes.
Yeah.
Finding people. Yes. I think we're done. So, thank you.
Thank you.